So I would like to, first of all, thank the organizers for giving me the opportunity to talk at this Open Docs meeting. And I'll be talking a little bit about the issues and solutions related to standardization of bioinformatics analysis and omics-based safety assessment. And to start off, I would like to just point out that systems biology, systems toxicology is essentially like any other science. So you have a hypothesis that you want to test, and you have an experimental design that you use for testing your hypothesis. And the main difference is that because of high throughput and high dimensional technologies, you're able to test many more hypotheses at the same time. And this leads to issues with data management, with essentially with multiple testing, and I'll be talking a little bit about that, and related to bioinformatics and chemoinformatics, which was discussed in the previous session. Okay, so what is bioinformatics analysis, and how do we sort of evaluate it? So if bioinformatics is like also a science like any other, then each analysis could be seen as an experiment. And so all the analyses that are performed should be, of course, valid and supported by literature. So don't invent any completely new methods unless you really have to. And the procedures and results should be made publicly available, and computer code also documented and recorded, and all the parameters, etc. And then raw data submitted along with the publication. And each analysis also needs to be controlled with positive and negative controls. So in omics analysis, or in general, it can be difficult to have, if you are investigating something completely new, it can be difficult to have positive controls, so you don't necessarily, but usually it can be good to repeat some other study which has been done before and which you really want to compare your results against. And then when you're making your conclusions, then it's a good idea to apply evidence from different tools, so not just to use one analysis tool like gene ontology, but also include other pathways or analysis tools like cake or reactome, etc., or even commercial tools like ingenuity pathway analysis, and then to use also different statistical methods. So if you're doing pathway analysis, you can use essentially different statistical tools to determine whether a pathway is significantly enriched or not, and these sorts of things. And bioinformatics, like any other science, is also to a degree an art, so it's important to take that into account as well, and to realize that you may not have completely standardizable workflows and so on. So anyway, this is a kind of a background towards what systems toxicology is all about. So it's a review from Kohonen et al., who is the director of NIH. And what this is depicting is that part of the attractiveness is that you can really increase the throughput of experiments. So from human epidemiological studies, maybe you can do 1 to 3 per year. And then sort of standard large-scale animal rodent studies, a bit more. And then if you're using more high-throughput alternative animal species, you can do even more. But it's very difficult to beat the throughput of in vitro assays, but they also come with additional caveats, and often it's more difficult to relate evidence from cellular models to organism or organ level phenotypes, or especially to population level phenomena. And one important way to do this, which we'll be discussing from bioinformatics point of view, is that you identify activities of so-called toxicity pathways, which are just biological mechanisms that are important for toxicology, and then you use computational biology to prioritize maybe some compounds for animal experiments, more or in some cases even to predict toxicity directly from them, which would be the ideal situation, of course. And to facilitate this extrapolation of results from cellular to organ and organismal level, this adverse outcome pathways concept has been developed. And omics studies, as shown in the middle, generally generates data of cellular responses, which can be predictive of exposure or molecular initiating events as a biomarker. And it can be also used to predict organ and organism responses. And this is essentially what we like to do. And by constructing evidence and arranging it in this sort of adverse outcome pathway, this is what toxicologists eventually hope to do. And this is a slide that we saw with Roland at the ECA meeting for new methods development, new methods for toxicological assessment. And we were really excited by this because it includes gene expression data at the top, the tier 1. And it makes sense from a toxicological point of view because the idea is presumably that you do first a very broad mechanistic assay where you can assess the presence or absence of many biological mechanisms at the same time. And then depending on the outcome of the assay, you design further experiments, for instance, experiments that test certain modes of action or key events in more detail using assays which are maybe more predictive or use more very sophisticated cellular models which you might not be able to use for everything otherwise. And this is a conception of how you analyze omics data. So essentially you have the experimental design, and we'll talk a little bit about the number of biological replicates. And you have pre-processing, and we'll talk a little bit about differential expression, and then touch briefly on pathway analysis and these sorts of methods. And there are some reviews under the slide which are sort of defining reviews, but starting with microarrays and more recently RNA sequencing. And this is just a brief reminder of what we're doing when we're doing this analysis. So extracting biological information from omics data using statistical methods, and the most simplest way to do this is to use in a treatment control situation is to use a t-test, but there are several complications to this which we will get to briefly. And in general, the problem with omics studies is that you don't have sufficient numbers of biological replicates to do proper statistical testing. And essentially the whole of bioinformatics is related to developing methods which work even if you have are doing too many parallel assays and too few biological replicates for standard statistical testing. And this is kind of a brief reminder of what kind of statistical methods exist. And I suppose if you're in the QSAR field, you know this already, but generally you have supervised and unsupervised methods. So unsupervised methods include clustering, principal component analysis, and such, which are used to discover new things about the data and new structures, and might be especially important when you're analyzing data to see if you have unknown experimental factors that you would need to take into account in the statistical analysis when you're building a linear model for the statistical analysis. And then if you have sufficient amounts of data, then you can use also supervised, or you can also use supervised methods. So these are the more traditional machine learning type tools, and also include, I think, in my opinion, t-test, because they also know which is treatment and which is control in general. And here are some examples of using unsupervised methods, and I would point out this graph especially. So this is a quality control situation where you have 3 different biological sample donors, and you can see from the PCA plot, and then you have a couple of different treatments, which are, here, some of them are completely on top of each other, and then huge separation between the different biological donors, so there's a lot of variability, especially in samples that come directly from humans or animals. And in this case, I was able to make some statistical conclusions on the data by taking this sample structure into account in the analysis. But obviously, this is going to increase the amount of noise that you have in this analysis. And so it's important to use the proper tools to do these analysis, and I would especially point to anything to do with bioinformatics. A very good source of tools is R bioconductor. So R in general is a statistical programming language, and one of the sort of founding packages of bioconductor is the lemma, and it has been specifically developed to deal with small sample sizes and to increase the power of the specificity of analysis, even if you don't have huge amounts of biological replicates. Okay, and it's doing this by engaging, for instance, in information borrowing. So it's using Bayesian methods to sort of try to figure out what the general gene level, variance level is as a function of measurement intensity, so mean variance modeling, and also including the sort of quality control parameters directly into the linear modeling approach and other things. So I think it's, in general, it's a very good idea to always try to use these types of tools when even when developing new bioinformatics methods, maybe for dose response data, because they have been designed to essentially deal with the central problems that bioinformatics data analysis is faced with. So this is then exemplified. So another way to deal with the issues with bioinformatics data analysis is to do more biological replicates, and there's been some. This is a very important question for experimentalists, because obviously doing biological replicates is very expensive, and the picture is a little bit bleak in that sense, because essentially this is there are a lot of results which are saying that you should have maybe at least 6 biological replicates, and if you really want to have a very high power in your experiment, then you should have maybe 12 biological replicates, which for many biologists is not achievable, but 3 to 6 would be something that is sort of truly recommended by multiple different analyses that have looked into this problem. And there are also differences between tools in terms of power. So I talked about lemma before, and lemma has generally very good characteristics. So in this slide, 3 pluses means that it's always very favorable outcome. Or at least as favorable as can be. So if we're looking first at the false positive rate, then there are a number of tools which are giving fairly favorable outcomes with small sample sizes. So these include sort of Welch t-test and significance analysis of microarrays, which is a kind of a classical method, and lemma. But then if we are looking at the power, then it becomes more problematic. So because of the various modeling strategies, lemma tends to have higher power at smaller sample sizes than these other tools. And this is another sort of very important question, which is kind of vexing for bioinformatics analysis, and this is the multiple testing issue, namely because if you have a microarray, you're generally analyzing expression of 20,000 genes, but if we, for a moment, imagine that we're only doing 10,000 genes, then if we're using the regular p less than 0.05 criterion for the statistical significance, then we would get from this array experiment 500 false positive discoveries, which sounds quite a lot, especially if we have a very fairly low dose treatment, which would have maybe 100 true positive differentially expressed genes. So in that case, we would have 85% false discovery rate, and which I think most people would agree is unacceptable from a scientific point of view. So some of the corrections that then have to be applied to this data include the false discovery rate correction, and the Q value test, this correction is commonly applied, and essentially it reduces the p-value significance threshold so that you have approximately 5 to 10% false discovery rate, which is thought to be sort of fairly acceptable by the community and represents maybe a sort of a realistically attainable biological standard, because you always have also unknown problems that you can't foresee in biological experiments, which reduce the level of replication of the results. But this kind of thinking can be taken even further. So you can essentially think of this reproducibility crisis to some degree as a science-wide multiple testing problem. So if you here the problem is that you don't, unlike in the omics experiment, you don't really have all the negative results, so you can't directly estimate the false discovery rate or the p-value that would be needed in order to overcome the false discovery problem in this sort of science-wide context. But there have been some attempts to do that, and they've sort of an estimate of the p-value which would be significant, even given given some realistic assumptions, would be somewhere like 0.005 or something like that, so 10 times lower than the regular 0.05 p-value. But this wouldn't probably solve all of the problems, but one way to improve this situation is to do more replicates, which would or improve the quality of the biological data, which would reduce the false discovery rate. So as a kind of a summary of that approach, so if you have a question where a situation where you have like a large random error, then the only thing that you can really do is increase the amount of biological replication. And if you have a situation where you have a large systematic error, this can be more problematic to overcome, because maybe it's very difficult to distinguish a situation where you, from the best possible situation, which would be this one, because if you don't have a point of reference, so then doing interlaboratory testing or additional controls might be very important, and trying to detect so-called batch effects and trying to normalize them with bioinformatics methods and so on. There are some references at the bottom. Or, of course, you would have a situation where you have a problem in both ways, but so then you would need to apply both types of solutions to that problem. And in general, this is a text from American Statistical Association about how to interpret data, p-value based results. And I think the summary would be that essentially you have to think biologically about what you're doing, and this is really the only solution. Don't trust the p-values, but also think about the biological significance in a wider context. And p-values are just testing the hypothesis that you formulated. It's not testing all the possible hypotheses that you could formulate and so on. So You can maybe access that link later. But as a sort of more specific set of solutions. So first of all, the problem that since you don't generally have very large amounts of biological replicates, omics studies tend to have low power. So they can give, if you repeat a low-powered study, you will most likely get slightly different results. And also, if you analyze the low-powered study with different bioinformatics tools, you will also most likely get slightly different results. And especially if you have low dose treatment, this can be a problem, but it can be alleviated to some degree by pathway analysis, because low-power study means that you are essentially randomly selecting from all the possible results that you could be getting. So pathway analysis, because of pathway redundancy, so you have usually many genes that are annotated to a single pathway. So if you do pathway analysis, you would be able to detect activation of a pathway even from incomplete data. And gene ontology is an example of pathway annotation system, so which we won't really go into, but essentially grouping genes by biological function and then doing statistical analysis on the groups. And as a further solution, which Ulla will maybe talk about, is automated pipelines. So essentially, if you don't really know which tools you should be using, or you have differing suspect that they might give somewhat different results, then if you run many different tools in parallel and then compare the results, you can get some sort of a better idea of what the most likely, what the variability is in terms of the results, and which results you should trust and so on. Okay. So this is the conclusion side. So bioinformatics is an integral part of increasing importance in systems toxicology for translational discovery, and workflows for standardized procedures are key to increase reliability. Our bioconductor framework is a very good set of tools that can provide solutions for many of these problems. And replicates are very advisable, and diverse tool applications increases reliability of data analysis. Okay.